All right, everybody, this is your last video in the section on experiments. This video covers blocking mostly, and at the end, I'll talk about inference a little bit. Okay, so blocking, just in general, is very similar um, to stratified sampling, right, in the way that um, in the groups that it makes. Okay, so oftentimes when people are designing experiments, they know that certain groups may have different reactions to the treatments that are imposed. So their response might be different depending on this one variable. A really common one is gender. People like to block by gender often. Um, for example, there are some medicines that are known to work differently and have different effects on women versus men, people who identify as male and people who identify as female. Um, Another common one is age. So sometimes if we, as the people who are designing the experiment, think that age might be a factor or might affect how the subjects respond to the treatment, then what we need to do before we give them the treatment is we need to block. We need to block by that variable, whatever it is, to eliminate any effect that that particular variable might have upon the response, on the response variable, the outcome of the experiment. So a block, okay, is a group of experimental units. Remember, if they're people, we call them subjects, that are known before the experiment to be similar in some way that could affect the response to treatments. Okay, so the blocks, because they are made up of groups of people that have something in common, right? They're similar in some way. That's what I mean by they are similar to strata, or you think about stratified random sampling, right? But strata and stratified random sampling only happens in observational studies. Blocking is what we do in experiments. Okay, so in a randomized block design, we're incorporating blocks into our already established completely randomized design. So rather than random assignment occurring right at the start, when you have the full sample, like the full group, and you would split the groups into 2 or 3 or 4, however many groups you needed for the number of treatments you had, rather than doing that, you split people by the block first, and then you do a completely randomized experiment within each block, and then combine the results at the end. So in a randomized block design, random assignment occurs inside the blocks. Okay, and I'm going to show you how to draw that in just a second. Okay, but when we draw them, you have to split the subjects into blocks first, right, to eliminate the effect, the possible effect of that variable, right, and then randomize within each block. And then you just have to remember to combine everything at the end, okay? There is one particular type of blocking called matched pairs. That's a very special type of blocking where each block is a matching pair of experimental units. Okay, so some examples that I gave you here were tires on a car. So say you wanted to test the wear and tear of an old tire brand versus a new tire brand. Well, if you do that on different vehicles with different drivers who live in different areas, right, that would mean there's a lot of confounding variables that you don't have any real control over. But if you put the old tires on one side of the car that you picked at random, right, either left or right, and then the other set on, like, the newer set on the other side of the car, right, then all of those confounding variables would be accounted for because the pair was the car, right? That each car would get both treatments, right? A really common one that people use when they're designing studies is giving a person both treatments in a random order, right? So, for example, maybe you wanted to know the effect of a certain sunscreen on um, on blocking UV rays, right? Um, People have lots of different skin types. They live in different areas, even within the same city. So there's lots of possible confounding variables there. You could have someone say, where, like, assign at random, um, you have a control, which is just like a cream with no actual sunscreen in it, and then you have the actual sunscreen. Or maybe you have an old version of the sunscreen and a new version of the sunscreen, right? Whatever you were trying to test. You could have assigned people at random to wear the control and then the new sunscreen for like a week and then switch. But the problem with that is that it could be, maybe that first week was a lot sunnier than the second week. So that could be a problem, right? So what would make the most sense is to have the person, split their face in half, right? And say, okay, put this, cream on the left side of your face, put this cream on the right side of your face, and then come in after a week and we'll, measure some stuff, right? And obviously, which cream goes on which side would be assigned at random. Okay, so that's matched pairs. You have to be able to literally match up the treatments or to one experimental unit or to like one person, one item, one subject, right? If you can't do that, then it's not matched pairs. Okay, so I gave you an example of what a block might look like. Okay, so this was an experiment about cell phone types. So you had 10 volunteers. So they thought that smartphone users versus non-smartphone users would have different responses to this. So they blocked by smartphone user first, then did random assignment within each block to split between the two treatments, which was you got a different type of keyboard for each, right? Keyboard A, keyboard B, right? Then you compare and then combine the results at the end, right? So basically you have two completely randomized designs going on at once, one within each block. Okay, so I'm going to pause the video here for a second. I want you to give this a try, see if you can draw. Oops, actually, let me go back one. One other thing I want to note is that they have the sample size, right, or the size of the group underneath. That's important, especially if you have uneven groups, right? See how here you've got only 4 in this group and 6 in this group. So important to note that when you draw it yourself. All right, so pause the video here. I want you to give this a try, okay? I know this is talking about ads you watched at the beginning of class. We're going to do an activity with this. So you may do this before we do that activity, but there it is. So I'm telling you that we're blocking by gender because we think that people are going to respond differently to these 3 different TV ads. Okay, so give this a try to sketch it out, and then come back and we'll see how you did. All right, so you can see on the right side of your screen, here is a randomized block design for 3 different ads blocked by gender. So we started with our 300 subjects at the beginning, then blocked by gender first. That's always the first step. So you have a block for women and a block for men. Notice that I gave a sample size for each one. Then random assignment comes next. Remember that if you are asked to do this on a free response question, you have to describe exactly how the random assignment would take place. So number the women from 1 to 120, randomly select the first a third of them using a random number generator to be in the group for ad 1, then another third for ad 2, the remaining would go to ad 3. Okay, and then you would also have to describe that for the men. But what you can do is just say, repeat the same process for the block of men, but putting 60 people, since a third of 180 is 60 people in each group rather than 40 people in each group for the women. Okay, then after you have the actual treatment that occurs, which is watching the different ads, they all come back together to compare. So we're comparing reactions to these ads, right? And then like within each block, and then at the end, you have to combine the results. Okay, so the real moral of the story with blocking, and really with all of this that we've been talking about, is that you need to control what you can. Okay, so that's in your experimental design, right? Do as best you can to control as many confounding variables that you can think of. Block on what you can't control, right? Oftentimes we can't control the age or location or gender or occupation or lots of things that we can't control, especially when people volunteer to participate in experiments. So if we think that those variables that we can't control are going to affect our outcomes in some way, we need to block on those variables. Okay, and then last thing is to randomize, create comparable groups. Okay, so that's the treatment groups, this stuff with like ad 1, ad 2, and ad 3, right? And the random assignment. All right. So very last thing that we need to talk about in this video is inference. Okay, so that's kind of the whole point of doing studies and experiments, right? We take all of these samples and we do these surveys and we do these experiments, but it doesn't matter much if we can't take what we've learned, or the truth that we've garnered from that sample, and apply it as best we can to the entire population of experimental units, right? So no matter what that is, typically we're talking about people, right? Because that's usually the most compelling truth, is the one about people and human behavior. So we have to be able to infer things from the sample about the population, right? But we can only do that if our experiment and our study was designed well. Okay, so there's 2 ways that you can infer things about the population. So if you have random selection of individuals, that means that you can expand your results, right, and make an inference about the entire population that those individuals came from. Okay, so basically saying you can't have volunteers. Volunteers is not random selection of individuals. Okay, if you randomly select your individuals, then we would say that because of the natural variation of the universe, that your random selection of individuals is likely a good representation of your whole population. So the conclusions that you draw about the sample likely are also true for the population. But that's only because you randomly selected the people um, or items, subjects in your study, right? So this is most common in observational studies, right? Then what we've been talking about in this section is random placement into of subjects into groups. Okay, that is when you can infer things about cause and effect. Okay, which is most common in experiments. Okay, um, and again, we talked about this, I think it was the last video, that the random placement of subjects into groups, the point of doing that and why it's included in our principles of experimental design is that we're trying to create equivalent groups to balance effects of other variables, right? And then if we have enough people, then we can distinguish the effects of the treatments from just those chance differences, right? So the random placement of subjects into groups is what allows us to infer things about cause and effect, that our treatment is actually what's causing the changes that we see in the response variable and not just random chance. All right. So that is it, everybody. I hope that all of this kind of like came together at the end and made some sort of sense. If you have any questions, let me know, and I'm here to answer them. So I will see you all in the next video. Bye, everybody.